How as an immunologist, I'm going to talk to you today actually about environmental issues and to do with stem cell transplantation in corals. And I will try to explain to you the idea that we're doing in our laboratory. So as an immunologist, I'm working on different organisms through collaborations. We had projects on tunicates, on tuna fish, we have with sea urchins, even snails. But today I will talk to you about a project we have in cnidarians, which are mainly, we're going to talk about hexacorallians, which are sea anemones and corals. And before I will start, I want to give credit to the very talented students and my lab manager who are working on this project. So it's all started when I met with Professor Nikki Traylor-Knowles, when she told me, I get different genes of immune system in corals. Do you think we can isolate the immune cells in corals, which was never done before? And together with her previous student, Grace Snyder, we actually isolated different cells from corals, and we used different methods of isolating cells, which are not really classical methods. But amazingly, we succeeded in isolating different cellular populations from stony corals, including phagocytic immune cells that are capable of engulfing bacteria, as it's shown here on the left. So the project I will be talking today about is actually the idea of developing stem cell transplantation for corals. And bear with me the thought. The idea is to isolate stem cells from a heat-resilient individual and transplant to a heat-sensitive one to create a chimera. And when the next heat wave will come, this coral will have a chance of survival. So this is the project we're developing. It is funded by the European Research Council and also Revive and Restore. Before you will have expectations of us doing transplantation in corals, we did a proof of concept in sea anemones, a Hexacorallian, close species, and now we're moving also into corals. So I will give you the example of the proof of concept we have done with the anemones. So we use transgenic anemones that we can follow those transplanted cells within the animals. And now we can know whether transplantation is successful or not. So the first question is, how do we prove that we have stem cell population when we do a transplantation? So we want to show that the cells can integrate and proliferate within the animal. They can differentiate to different cell types within the animal. They can rescue animals from little doses of chemotherapy or radiation. Of course, those cells need to have longevity for them to work in therapies. It should be a specific subpopulation of cells, and of course, persistence of the genes and the phenotype where they're coming from. This is the whole base of therapy to work in the future. So I hope I will convince you that we have found all of those parameters in those Hexacorallian species. So we have done transplantation and we can find those transplanted cells. We get up to 40% of chimerism of the cells coming from the original donor 2 months after transplantation. We can find also the genes through PCR of the transplanted cells and their tissues. We can also rescue animals from little doses of chemotherapy by doing transplantation, showing that the cells are integrating and function in the animals. For longevity, what we have done is serial transplantation analysis. And the idea here is that we have done one transplantation, then 6 to 8 weeks later, we isolated again those red cells and did again transplantation. And then after a month and a half, we had again integration of those red cells into the animals. To look for specific subpopulations, we use this unique marker of aldehyde dehydrogenase in combination with additional markers to find those small round cells that we suspect as our candidate stem cell population. We have done those transplantation, and with very low amount of cells, we succeeded in getting chimerism with this transplantation. We also showed that those cells after 2 months of transplantation are differentiating from those small round cells into the diverse cell population within those sea anemones. And I hope I convinced you that we succeed in showing our candidate stem cell populations through different functional assays. And what is unique that we have done is that those isolation methods are not specific for a species. So we can use it through different species of Hexacorallians, including stony corals. And now we're moving into the stony corals to actually do this transplantation. You can see here Alona, my student, doing this transplantation in corals now, testing our hypothesis to see whether we can develop this tool of stem cell transplantation for rescuing corals. So I would like to thank, of course, my collaborators, my laboratory, our funding sources, and thank you for listening to me. If you have any questions, that would be a good time.